we are ready to proceed with the most challenging, I would say, for the students to understand stage of this process, such as the stage involving steps 6, 7, and 8, which involve C-C bond cleavage and formation. And in this stage, we are going to use new enzymes for us. They are called transketalase and transaldolase. But let's first pay attention on the stoichiometry of this reaction. And another kind of thing that I wanted to mention, that actually this stage happens only when ribose 5-phosphate is not efficiently used for nucleotide biosynthesis, and those carbon atoms are required by the cell for energy purposes, right? In this case, they need to be converted to those glycolytic intermediates, such as fructose 6-phosphate and glyceraldehyde 3-phosphate. Okay, stoichiometry of the reaction. So we started with 3 molecules of glucose 6-phosphate, right? So this oxidative stage produces 3 molecules of ribulose 5-phosphate. Each of them contains 5 carbon atoms, and 3 molecules of CO2. So that's our oxidative stage, steps 1 through 3. So now let's cover CO2, because we are going to focus on just ribulose 5-phosphate molecules and their fate. So 3 molecules, 3 ribulose 5-phosphate molecules gets isomerized into something. So 2 of them are isomerized in, they undergo epimerization, right? They are isomerized into xylulose 5-phosphate. And only 1 of the 3 is isomerized into aldose, such as ribose 5-phosphate. So here we have isomerization reaction. So next, those carbon atoms get rearranged so that to form 2 molecules of 6-carbon fructose 6-phosphate and 1 molecule of glyceraldehyde 3-phosphate. So here we have this last stage, 3 steps, the remaining 3 steps that will be C-C bond cleavage and formation. So transketalase and transaldolase catalyze those reactions that require for steps 6, 7, and 8 of the reaction. Transketalase transferred 2 carbon units, and it requires TPP cofactor, as we will see on the next slide, and it will catalyze 2 steps in this remaining 3 steps of non-oxidative stage, so steps 6 and 8. Transaldolase resembles mechanism of simple aldolase, but instead of just cleaving the bond, it transferred the 3 carbon units from one monosaccharide to another. So since it's similar to aldolase, it utilizes similar mechanism that involves protonated shift-based intermediate. And we will see the action of transaldolase only once in the pentose phosphate pathway, which will be step 7. So what's the difference? Why 2 enzymes are required for C-C bond cleavage? So transaldolase catalyzes cleavage of the C-C bond between alpha carbon with respect to the keto group and beta carbon, right? So this bond is going to be cleaved. It utilizes a lysine residue of the active site amino acid side chain, right? In order to form this protonated shift base that serves as an electron sink in order to delocalize the electrons from the cleavable C-C bond. In case of transketalase, the cleavage occurs between the carbon of the carbonyl group and alpha carbon. And so it needs to rely on external electron sink, such as the one that is provided by thiamine pyrophosphate or TPP cofactor. So here we have this electron sink that helps to delocalize the electron density that is assigned to that portion of the molecule upon cleavage of the C-C bond. So we will start with transketalase and its cofactor. We saw it already before for decarboxylation of pyruvate, right? For yeast fermentation, for alcoholic fermentation. And we know that this thiazolum ring of TPP is characterized by highly acidic proton that allows TPP thiazolum ring to be converted into the corresponding E lead, which serves as a good nucleophile. So it can attack the electrophilic carbon of the carbonyl group, forming this intermediate And here we have polarization of the CC bond. Again, this was our alpha carbon before. I mean, sorry, a carbonyl carbon before, and this was our alpha carbon before, right? And here is the bond that is being cleaved, right? The bottom of the substrate of this reaction, xylulose 5-phosphate, is released in the form of glyceraldehyde 3-phosphate, and the top portion, those two carbon atoms that will be transferred to another monosaccharide, they are still attached to TPP in a resonance stabilized carbanion. Again, here we have a nucleophilic carbon because it's negatively charged, so it can attack the electrophilic carbon of the aldehyde group of another substrate of this reaction, ribose 5-phosphate. First, similar intermediate as before is formed, right? But then, due to the polarization of this bond, the carbohydrate portion attached to TPP can be released in the form of a ketose. So as a result of the reaction between xylulose 5-phosphate and ribose 5-phosphate, we form pseudoheptalose 7-phosphate and glyceraldehyde 3-phosphate. So the alpha carbon of the original xylulose 5-phosphate, the ketose that enters the reaction first, will be the first carbon, actually the carbon of the aldehyde group of glyceraldehyde 3-phosphate. So pseudoheptalose 7-phosphate and glyceraldehyde 3-phosphate will both proceed to step number 7 on the pentose phosphate pathway, which is catalyzed by transaldolase. So transaldolase again resembles aldolase. It also has this catalytically active lysine residue in its active site. First, it attacks the carbonyl group of pseudoheptalose 7-phosphate, the ketose out of the two, to form this covalent intermediate protonated shift base, which helps to polarize the bond between alpha and beta carbons, right? Alpha and beta carbons, and deprotonation of the beta hydroxyl group causes the bond between alpha and beta carbons to be cleaved and release an aldose containing 4 carbon atoms, the bottom portion of pseudoheptalose 7-phosphate, so that the beta carbon of the original pseudoheptalose 7-phosphate becomes the carbon of the aldehyde group. And the top portion, the top 3 carbons of pseudoheptalose 7-phosphate are still attached to lysine, and here we have those this carbanion, which is resonance stabilized. It's nucleophilic. It will attack the electrophilic carbonyl group, aldehyde group of glyceraldehyde 3-phosphate, to produce fructose 6-phosphate, right? To kind of transfer those 3 carbon fragment, this 3 carbon fragment on top of glyceraldehyde 3-phosphate to form fructose 6-phosphate. So again, we started with 7 carbon ketose and 3 carbon aldose, and as a result, we formed 4 carbon aldose and 6 carbon ketose. And erythrose 4-phosphate will be further used in the last step of the pathway, but fructose 6-phosphate is already the product. It's already released, and it can be used. It can enter glycolysis if needed, or it can be rearranged back into glucose 6-phosphate to keep cycling through the pentose phosphate pathway. So let's take a look on another look on the mechanism of transketolase for the last step of the pathway. The same elite of TPP attacks now the keto group of xylulose 5-phosphate, the remaining one. Remember, we converted 3 molecules of ribose 5-phosphate into, ribulose 5-phosphate into 2 xylulose 5-phosphate molecules and 1 ribose 5-phosphate molecules. We used one of the 2 xylulose 5-phosphate molecules, and this is the remaining one. So again, the same product, the same intermediate is formed. Polarization of the bond between carbonyl carbon and alpha carbon helps to cleave the bond and release glyceraldehyde 3-phosphate. The resonance stabilized carbanion is nucleophilic. It would attack the carbonyl group of erythrose 4-phosphate, and this will result in the formation of the bond between this carbon and the carbon, negatively charged carbon of this intermediate. And after abstraction of proton from this oxygen, the bond between carbon and the carbon of TPP moiety can be cleaved, and the second fructose 6-phosphate molecule will be released. So here we have both compounds that are no longer used, right? They will be entering glycolysis, or again, there are some ways how they can be rearranged back into glucose 6-phosphate to be cycled through pentose phosphate pathway. So again, that's a summary of stage 3, the most difficult stage of the pathway. So step 6 involves the reaction between xylulose 5-phosphate, and that's a ketose, and ribose 5-phosphate, which is aldose, to produce pseudoheptulose 7-phosphate ketose and glyceraldehyde 3-phosphate aldose. Transfer of 2 carbon units is catalyzed by transketolase. Then the product of step 6 become reactant of step 7. So again, now we have transfer of 3 carbon units from top of this ketose and placing them on top of this aldose to form fructose 6-phosphate, a ketose, and erythrose 4-phosphate, aldose. Reaction is catalyzed by transaldolase. And finally, the remaining molecule of xylulose 5-phosphate reacts with one of the products of step 7, erythrose 4-phosphate, again ketose, aldose, form ketose and aldose, catalyzed by transketolase. And here we have the net reaction for this stage 3 reaction.